So my group is interested in understanding the signal transduction pathways that regulates how cells can sense and migrate directionally when exposed to external chemical gradients. That's a process that's known as chemotaxis. So when you expose a cell to an external chemoattractant gradient, the cell responds by acquiring a polarized morphology. In a paper that is published in this issue of Developmental Cell, we set out to determine the role of the target of rapamycin in regulating neutrophil chemotaxis. The target of rapamycin is a serine-threonine protein kinase that integrates external signals to regulate a variety of cellular processes. The mTOR in mammalian cells is actually placed in two complexes. So one of them is called mTORC1 and the other one is called mTORC2. And what we ended up wanting to do is determine the role of mTORC2 in the control of neutrophil chemotaxis.
To inhibit TORC2, we knocked down one of specific component of mTORC2 receptor in neutrophil neck cell. We found the receptor knockdown dramatically inhibit the neutrophil chemotaxis. It appears receptor knockdown cell can sense chemoattractant stimulation, but they cannot be fully polarized. Indeed, we found receptor knockdown cells F-actin is uniformly accumulated around the cell. Interestingly, we found neutrophil chemotaxis is especially dependent on mTORC2 for inhibition of mTORC1 does not affect neutrophil chemotaxis. It's also not dependent on AKT, main downstream effect of mTORC2.
So let's see what do we learn from dictyostelium as a model organism. So as shown in this cartoon, the PI3 kinase signaling and TORC2 signaling is essential for adenylyl cyclase activation and cyclic AMP production. So we wanted to look like whether this pathway is conserved in neutrophils. For this purpose, we checked the chemoattractant mediated cyclic AMP production in Richter knockdown cells. And as evident in this graph, you can see the cyclic AMP levels specifically absent in these cells. And then we wanted also to look the role of cyclic AMP in mTOR function. For this purpose, we have proposed earlier that AC9 is the predominant isoform of adenylyl cyclase in neutrophils. We therefore knock down AC9 and then we check the chemoattractant mediated cyclic AMP production, and as evident in this graph, the cyclic AMP levels are much lower in these cells. Further, we wanted to determine the role of cyclic AMP in mTOR function, and we looked at the chemotactic ability of AC9 knockdown cells. As shown in this movie, the AC9 knockdown cells, although they can sense the chemoattractant gradient very well, but they have severe back retraction defect.
Tail retraction is regulated by ROA ROCK signal pathway. This pathway leads to myosin phosphorylation and then regulates back retraction. As AC9 knockdown cell shows back retraction defects, so we check myosin phosphorylation and the ROA activity in these cells. We found both in AC9 knockdown and Richter knockdown cell, they have higher myosin phosphorylation level. They also show higher constant ROA activity.
So from these findings, we propose that mTORC2 mediates its effect to regulate neutrophil chemotaxis and polarity by regulating both actin and myosin 2 filaments independently. So while the mechanism by which mTORC2 regulates actin filament assembly is not really understood, we now propose that mTORC2 regulates myosin 2 filament assembly using ROA and cyclic AMP dependent pathways. And we envision that actually these findings will be expandable to other types of cells that also move quite rapidly, such as T cells and dendritic cells. And we also propose that perhaps these pathways are also at play during metastasis, when cells are differentiating and acquiring more amoeboid-like migration that is observed in neutrophils and as well in dictyostelium.